Okay, so I'll start out with presenting my goal. It's to find a small, efficient ligating ribozyme. And I hope by now you guys know what a ribozyme is. It's an RNA that has catalytic function. And why ligation? Well, if you're going to talk about origin of life studies, you want to try to generate more complex products. So your products should be more complex than your starting material. And that's what ligation is. Ligation is taking two pieces of RNA, you're putting them together, you're ligating them into one product, a more complex product. So I want to increase ligation in a ribozyme, and I want to try using different conditions, particularly by freezing. And then I want to increase that yield by introducing thermal cycling or freeze-thaw cycles. So I won't go very much into the background for the RNA world. I'm assuming that everyone knows this by now, this idea started around the late 1960s. And it suggested that RNA is the first biomolecule as opposed to DNA or protein. And it's because RNA can store information and it also has an enzymatic function. But one of the major problems for RNA is that it's fragile. And if you ask anybody who works with RNA, they'll tell you it's prone to degradation. So if you heat it, it'll probably degrade. And pray if you have something like magnesium in your sample. So one solution to that is lowering your incubation temperature. And I guess when a lot of people think about freezing or ice, they think about all the water molecules and all the solutes in solid phase. Well, that's true if you go below this eutectic temperature on this phase diagram here. What we're interested in is this triangle area here, where the majority of the water molecule is in an ice crystal, but the remaining liquid water is mixed with your solutes, and that makes up your eutectic composition. And this eutectic composition exists in channels, very small, micrometer in width, grooves, and it's shown right here, so very small tunnels in the ice. And this provides a protective environment for your RNA. It concentrates your solutes, and for your RNA, you could also reach alternative stable conformations that wouldn't be possible if you tried incubating at higher temperatures. And above all, your RNA can still retain some enzymatic function. This is demonstrated by the R18 polymerase. This is from a paper in 2010. The R18 polymerase is an artificial ribozyme. It's lab created, and it's outlined in black right here. And it binds to this template in green, and it adds nucleotides one by one to this primer strand right here. So when people think about polymerase, they think about a polymerase that's made out of protein. And a common one is this T7 RNA polymerase right here from bacteriophage. And it does wonderfully at 37 degrees. So the higher you see these bands up here, the higher you go, the longer the products are. But at -7, if you freeze it, you don't see any polymerization. So how does the R18 fare? At 37, it's not shown here. At 37, it doesn't do so well because the polymerase degrades. But if you start at a slightly lower temperature than room temperature, it does comparably well as the protein polymerase. But the major difference is at -7. So here, the R18 can still catalyze polymerization at -7, below freezing, where the protein equivalent, you see no product at all. So this is something that RNA can do that the protein can't. But this polymerase is a little too long. It's a little too big for what I want to work with. So this is what I'm actually working with. This is the Hammerhead Ribozyme, and this was first found in the mid-1980s in plant viroids. This specific sequence, though, is from Schistosoma, which is a parasitic worm. So this ribozyme is traditionally known to cleave and not ligate, even though they're both reversible reactions. So in a cleavage reaction, you would have the enzyme, which I outlined in red here, bind to the substrate in blue, and it would cleave it, or cut it into two pieces, generating two fragments, this P1 fragment down here and this P2 fragment up there. In a ligation reaction, the opposite would happen, where the ribozyme binds to P1 fragment and the P2 fragment, and it would ligate it back together, back to one strand. Efficient ligation with this sequence wasn't demonstrated until 2007. Here, the P1 fragment is labeled, and if it's ligated to P2, then you should see this band shift up here. And this was done at room temperature, but it was only possible if you added millimolar amounts of magnesium. And this is a fast reaction, so here, 18 seconds, you're done. And their maximum yield, 23%. So I thought this was a great ribozyme to start out with. I started out with magnesium, repeating their experiments, but I also tried calcium and iron. And in short, they worked. But that's not the focus of this talk, although you're welcome to ask me about it, iron people. So what I'm focused on is ligation through freezing. And if you do it this way, you don't have to have divalent cations in your sample. This is just a short example of the different temperatures I used. And I think that C is supposed to go there. But temperature alone isn't enough. So as demonstrated here, these two lanes right here, they're incubated at the same temperature, -10. But one was frozen before incubation and one remained liquid the entire time. So even though it's the same incubation temperature, you still have very different results. This demonstrates that you need freezing, or the concentrating effect, to get ligation. So next we looked at how fast this reaction is. And while it's not that 18 seconds from, that you get with magnesium, we still get a higher final yield. And based on this, the majority of ligation is done in an hour. So we decided to use 1 hour in our thermal cycling experiments. So thermal cycling is freeze-thaw cycling, and it's supposed to mimic day-night cycles. So each of our cycles there's a 2-minute heat step, and then there's an hour-long incubation freeze step. So that's one cycle here, and then 2 cycles, you have 2 heat steps, and then 2 hour-long freeze steps. And that wasn't supposed to happen. You should see like fans up here. So our base yield, what we started out with is 25%, but with each additional cycle, so basically each additional day, we should get more and more ligated product. So we thought this was a great start, but before going further into cycling experiments, we wanted to try increasing that base yield, that 25%. So we already briefly went over different temperatures. You can get between 25 to 30% depending on if you're between -20 or -10. We tried looking at sugars, millimolar amounts of glucose, sucrose, lactose, trehalose. Well, it didn't help, it didn't hurt. We tried looking at different buffers, PPIS, sodium phosphate, tris, and as long as they were the same pH, they were comparable. pH is important. So our standard is pH 8. And if you go a little higher, it didn't change the yield. But if you go lower, it'll start decreasing that yield. And if you go about 5.5, it just killed the reaction. There's just no ligation going on. Now, amino acids. Now, why would I be talking about amino acids if this is in the context of the RNA world? Well, let's face it. I doubt early Earth was as clean as our test tubes. We decided to look at what would happen if we added or contaminated our samples with amino acids. So we tried looking at the 20 standard amino acids. And I didn't list all of them, but 18 out of the 20 fell in this category here. None of them actually increased the ligation yield, but none of them dropped it below 20%. Only 2 of them drastically reduced our yield, which is tryptophan and isoleucine. And overall, I think this is good news because the earliest amino acids, at least the ones that are proposed earliest amino acids, are on this side of the list. And the last thing we tried was anions. This might sound a little strange because with RNA studies, you usually focus more on cation effects. And that's because RNA is negatively charged, cations are positive, so you would expect interactions. We didn't think that anions would contribute very much, but, well, we were wrong. So depending on which sodium salt you used, you get very different results. Our best ones were from acetate, formate, and citrate, and we think it has something to do with our carboxyl groups. Fluoride, chloride, bromide, and iodide. They seem to follow some general patterns, like fluorine and chlorine are supposed to be more electronegative than bromine and iodine. They're also smaller in size than bromine and iodine. And in the context of chemical abundance, there's a lot more fluorine and chlorine than bromine and iodine. And we also took this as another piece of good news because based on this, we don't want a lot of bromine and iodine around. We're still not sure exactly how the anions are affecting the ligation yield, but we are exploring some ideas. So that's where we are experimentally. I'd like to end this talk by summarizing that ice preserves RNA, and it also preserves the enzymatic functions of your RNA, particularly ligation, what we're looking at. And you don't need divalent cations to do it. And this ligation reaction also tolerates the majority of amino acids, the osmolytes that we looked at, and you can try using different buffers and different salts, and you can still get ligation. You can increase the base ligation yield by freeze-thaw cycles, and really this last point is our next step. It's to combine thermal cycling with our best ligation yield, and that was that 40% from acetate. So we want to get above that 40%, which is our best so far. So that's it.
That was really cool. Have you given any insight into how this might vary with pressure? Because both the panspermia hypothesis and the idea of delivery of volatiles to the Earth could mean that if RNA is kind of having fun ligating itself on comets for a long time, it could be safe there.
I don't know about low pressure, but I did look a little bit into very high pressure, and that could actually affect the way the ice is formed. So this type of ice, it's normal pressure, and it's phase 1H ice. But with high pressure, you get different types of phases of ice. I don't know. I haven't tried different pressures. It's just what was in our lab.
Cool, thanks.
Any other questions for lab? Nope. Yes.
So going off of the pressure in the ice bit, you were mentioning that this reaction only occurs under these frozen conditions where you have the solid phase, and you're attributing that to an increase in the concentration. Is there any reason to think it might be an effect of surface chemistry with the ice?
We don't know if, like, for instance, we don't know if the RNA is somehow like concentrating near the surface, the walls of those tunnels. We don't know. So far, all we know is that there is a concentrating effect, and you can visibly see that if you stain it, if you stain the RNA and you detect fluorescence. But no, I don't know.
Okay, cool.
All right, let's thank Lively again.